And you've seen this before, of course. This is the 3D viewer. But we haven't really covered that good enough, I think. And there are a few things that I wanted to show you. So first of all, very obvious are these anatomical slides that you can just drag around in space. But you can only do that if no other tool is selected. So basically, you have to click on this second button with the 4 arrows, and then you can move them around like this. And there's also this second small window down here, anatomy slices. And here we can also like put in coordinates. For example, if we want to set the x-plane to 0, which is the mid-sagittal plane, we just enter 0 and hit enter, and then it's in the middle, right? So that's another option. And what we can also do is just to hide them, right? We can say we only want to look at the electrode itself without any anatomy. Or there's also transparency. We don't use that much, but you could set this to 20%, and then this, or maybe let's say 50, then it's a bit translucent, right?
So we can change here to see the render, the naked space, the normalized space, and the equation space too?
Not in the render, you have to reopen it actually. Yeah, you would have to. So here, and then run. Yeah, but what you can change here is the backdrop. So you can say we want to T2 M&I space instead of or the proton density or the PCA. But you can also instead use the patient pre-op T2, and then it will show the patient-specific M&I space warped T2. And then there are also some additional things. For example, you can also import different files, like the big brain of Kathleen Amund's, or so other nifty files that you want to show here. And that is possible. I can quickly show you. You would need to put a folder into templates, into the space folder that we've covered before. Sorry. So into this folder, you probably know this folder by now. It's in Lead-DBS template space and then the M&I space. And you can create a folder called backdrops, and then you can put in nifty files that you can additionally specify. And so now we have, it took a while to load, but this is a high resolution histology image from Jülich, which we can also use to visualize as a backdrop, right? So basically any nifty file you want to show, if you have your own template or so, it should be an M&I space, but these you can put into a folder called backdrops in here, and then they will be shown. Okay, and that is that. Then what Friederike asked me also to check once more, so you have the stimulations that we've created yesterday, and these are stored, so the nifty files of these are stored in a folder that's called Stimulations. Wait a second, I'll make my screen size a bit bigger again, so you can read. And so we have the patient folder, which is this one, course example. Then we have stimulations, and then we have the name of the stimulation that you entered. And in here we have two files. One is called the BAT left and one the right, right? So these are basically just nifty files of the stimulation. So you can use them in any other software, like in tractography or SPM or whatever, right? So these are just normal nifty files that you can load anywhere. And you can also visualize them here by running this add objects OpenROY. So you basically can run any Nifty file, show any Nifty file here. So if I just select this write VTA, for example, I can give it a color, and then it will be here, right? So let's see. So this is now just the same, the VTA we created yesterday. Remember, Tanya wanted to use the lowest contact, and we just visualize the Nifty file. The other option, of course, would be just to reopen the one we did and press visualize, and then it should also, create it. And you see there's even some slight difference because of the thresholding, but it will give you essentially the same information. Right? So this is really good to know, I think, where these files are stored, because at some point you will probably use other software as well and combine it with Lead-DBS, and then it's really great to know, okay, the VTA files are in here, and we can use them. There are some more files, so we have copies of the VTA, basically, which are called E-field. These are not binarized VTAs, right? These are the E-fields as the FEM algorithm calculated it, and E-field means the distribution of voltage in space, right? So if we have a gradient of voltage that is strong, then there will be a strong white pixel here, right? So these are basically the unthresholded VTAs. Then we have also copies of these which are Gaussianized, so they can be used maybe for some other statistical methods or so. So that's that. Then maybe some more smaller things. Up here we have some buttons for lights in the scene, right? So if we declick these light buttons, then the scene will change a bit and the lighting will change. So sometimes if you make a figure for a paper or so, this can really make a difference. Like that, you see, so it's a bit different lighting. Maybe check on your computer so you'll see it better, but now it's all a bit darker. And sometimes depending on the scene, it may help to change the light. Maybe helpful. So you have 4 different lights in the scene and you can turn them each on and off. And what else should we show? That's fine. And all that. Yeah, here you have the labels of the nuclei, so you can also show these. That should work, I think. And yeah, now we see this is the GPI, GPE, STN down here, red nucleus. For example, here the red nucleus in red. It's all a bit slow now because of this backdrop that I loaded, but you can see the labels now here. And that's about that, I think. Any questions? Okay, there was even an error. Any questions about this viewer? Do you have any thoughts about it or anything we could change or? Yeah.
If we want to run tractography in another software, so we will use the Nifty, the Nifty, and which image should we use as our base space? The shell?
The M&I's template, I think. So inside the same folder, so again in this.
I will need the DTI file
of the patient.
Which one should I
Okay.
use?
Then
Normalize.
so if you do that, you have to convert the VTAs are in M&I space, right? So you would have to convert these to patient space first. You can do that with this tools map file from template to anchor space, right? So if I run this, I could now select the VTA. select that, and now it will create a warp version which will be in the T1 space. So it's this one here now. And now you see it's the bubble in T1 space. And then you could use whatever software to go to the B0 space from T1.
So sorry, that was in tools?
Yeah, that's in tools. Map file from template to anchor space and run. You can also export code that would do so, because then you can script your pipeline. But if you just want to create one warp version, then there is also this map file from template to untouched anchor space, which would be the original T1, the one before you imported it to Lead-DBS. If you have an FSL project where you didn't touch the T1 also, that should also work. But then you have to convert it to B0 space yourself somehow with SPM or FSL. Okay, great. Then maybe I can also quickly show you the microelectrode recording tool, because I know you were a bit interested in it at least. One thing about it is that I haven't programmed it, so I only know the basics. Sorry about that. But I can show you at least where to open it and how to look at it. And so I will have just opened the same scene again. Let's see, it's easy. And I will hide all of these files for now. And then if you click on this button with the flash, oops, some bug with my display, but this button, then you get the microelectrode recording control figure. And as you can see, there are now these 4 trajectories around the microelectrode. And best is probably to even hide the DBS electrodes for now. And then we see the 5 electrodes that we usually have in the microelectrode settings. And what we now can do is to say we enable the keyboard control, for example, the right central lead, right? So we can say right central is on, keyboard control down here. Now if we go to this figure and we use the arrows, then this moves, right? And we see, maybe I can, let's see, let's do like that. So if we move this, we also see here that the position moved, right? Do you see that? Now we are at 4 millimeters before target. So here, right? So if we move this, then we see we are at this point. Then if we just press space, then it will create a bubble at this point. Right? So this way we could go, usually in Berlin at least, we often start 7 millimeters before target. Then we could say maybe there was an SDN cell, another one, and another one, and then stop. And then we have some space mark here. And what I did is press space for these green bubbles, these yellow bubbles, but there are also some more commands. So I know that there's T for top, I think, and B, no. Okay, there is generic, okay, it's already marked. But if, so I think T for top of SDN would work. So T, if you press T, it's a blue bubble, and bottom would also be bottom of the SDN. And there are also some other functions, and I think these are even documented in the manual on the website. And so if you're interested in that, you can play around with it, but that's more or less what I know about it. So of course we can now say we switch to anterior now, and we move the other electrode. We say the top was here, and then we had some. We have some cells here, and then the bottom maybe was here. That way you can mark the electrophysiological orders of the STM. And you can store this data with this button here, save state, and then also load state. But I'm not exactly, so it's saved in this file, and I think if you just load it, will load the one that you saved before. And I think for the people with next frames, but we don't have a next frame, so I cannot really tell you much about this. There's this button up here, the N button, and in here you can enter all of these next frame settings. So this might be of interest for some people. Okay, so I think this is the most basic, or the most things from this viewer. There is also the connectivity visualization button up here, but we will cover that a bit later. And I think that's, for now, that's that. Are there questions, or are there other things you would like to visualize, or things you... So what you also can do, maybe one more thing, is if you add objects and add a tract, you can also open track-list files, so you can visualize fiber tracts. That sometimes is a bit complicated because of the spacing. So you would really need to make sure that the space makes sense, that it's in NI space in this case, or in the native space. And sometimes these files are not stored really properly, depending on the software you use, but you can play around with that at least. And yeah, that's that. Any questions? Thoughts?
The output when I run to, as you were saying, when I modify the Nifty file from the VTA, what should be to modify from?
It will be this one, so GL that left in the patient folder. Yeah. That should be exactly the same resolution as the T1s. One more thing that Friederike told me to show you is this folder. It's called the SCRF, which stands for subcortical refine, and that is basically where the brain shift correction is stored. Right. So if you ever have problems with brain shift or want to rerun it or something, then you can just delete this folder and rerun it. That's helpful to know, because if there are, if the normalization was wrong and then brain shift ran wrong, so you just want to rerun everything with brain shift correction, that's everything stored in this SCRF folder. Yeah, so that's fine. Right. So then I'll start with the first talk about Connectomic deep brain stimulation, and I want to welcome you back to the theoretical part of this workshop. I also wanted to quickly announce that we plan to make a group picture before the lunch break. So if you want to be on that, or it would be great to do that, then please join us for that. And I guess the theme for today will be more about connectomics. But if you think you still need more time on the other stuff that we did yesterday, please let us know, right? We can also recap more about normalization, about and they're basically moving in a Brownian motion fashion, so they're diffusing across space, but they are hindered by these two barriers. So it's called hindered diffusion, because they bump here pretty often and go back there, but only sometimes can escape the diffusion. And if we could, and so I'll start again. You've seen this and you've seen that. So it's not synced together. Okay, if we could measure diffusion in brain, you see these balls diffusing, you have hindered diffusion. So that means that there's more diffusion in this direction or in this compartment and in this compartment than in the whole. And maybe in this part there might be more diffusion along the way, just because the balls get kicked. They could go like this. And so this is hindered diffusion. They can escape, but they are more likely to diffuse this way. If we could measure this in the scanner, if we put a bucket of water into the scanner, diffusion would be the same in all directions, right? So we would get something like this: a sphere, probability to diffuse in each direction is the same, right? But if we instead put in asparagus, do you have that in China? Asparagus? Yes, I guess so. These plants.
The green ones here.
They have fibers with water inside, so they have hindered diffusion inside of them. And our MR physicist at the institute where I was before actually did that. So he put asparagus in the scanner a lot to do tractography in the asparagus. And then you would get something like this. So you have more diffusion alongside the tracts, right? More than in this direction. So you have a stronger probability that the water diffuses this way than this way. And in the brain now, we have very high organization coming from so very, very strong bundles that are already very good organized. But inside there, we again have a lot of organization in the same way. And then inside there, we even have more compartments. And in there, we even have more compartments. So there's all the different diffusion. The water is actually very organized in most parts of the brain. We have a strong hindered diffusion. So more diffusion in one direction than in the other. And you could see that the diffusion in this direction would be stopped by the membranes, but there's free diffusion along this direction, so you get stronger, more probability that the diffusion goes this way than this way. So, and inside the brain, so what we could do if we have voxels now, this is very, very small voxels, and it would be a dream to have these, but we have much bigger voxels, but still, we could then measure the probability of diffusion here, which is stronger in this direction than in this direction. And this is a bit technical now, but we actually can measure diffusion. And we do this, I won't go into details, but we do this by applying two pulses, a 90-degree pulse and then an inversion pulse. And so basically you can think about it of taking two pictures. You take the first picture and you tag the water molecules, and then if they diffuse away, if you take the second picture, then you get a signal loss. And so you get a black picture because they diffuse away. So that's basically the idea. So the more they diffuse away, the less signal you get in the end, right? And then we can change the way of tagging the picture, the water molecules. So we tag them in one gradient direction, but we can move this direction. And then we get pictures like this, where we have, for example, if we have a diffusion direction like this, we get a lot of signal loss in these regions because the water diffused away in this direction. This is the anterior part of the corpus callosum, so also called, this is the splenium, so this is the forceps major. Okay, so it's part of the corpus callosum, and diffusion in this direction is strong here, so we get a signal loss if we tag for this diffusion direction. If we tag for this direction, we don't get signal loss here, right? So we can take different images with different diffusion directions, and depending on how the water diffuses, we get more signal loss or less signal loss. The idea now is to take a lot of these pictures, right, to say we take 12 directions, or even go crazy, and this is rarely done, but maybe it's something like this or this is more often done, to take a lot of directions, and we get a really good image. So in each of these directions is a whole rain picture, right? And we get signal loss where water diffuses away in this direction. And then we can go to the tensor. I skipped that, I guess, but in the end we will fit, or in ancient times we did fit such a simple model which said there's more diffusion in this direction, a bit less in this, and less in this. Right? So in comparison, this is a T1-weighted image, and each voxel, each pixel here, is just one number. So it says maybe 20 or something, right? So it's one number, nothing else. In the diffusion tensor imaging, DTI, we have in each voxel we have an ellipsoid. So we have a whole complex object basically that's defined by a few numbers that says there's a lot of diffusion in this direction and not so much in this direction. And then we can build the whole brain with that, right? So we will have these tensors, so these ellipsoids, and here you can see the corpus callosum that goes like this, so into the image. So it should be, the ellipsoids point in this direction. This is the cingulum bundle where we have the ellipsoids pointing in this direction, and we can then use this information to reconstruct fibers. Right? But every time you use stratigraphy, it's not magic, but it's basically a very indirect measure of water diffusion. And it has a lot of limitations, so you should never forget that. It's really a very, very indirect measure. But it's still the only one to do it without opening the skull and cutting things. Okay, so this is a two-dimensional set of tensors now. And we have, maybe you can see there's a strong direction here, so there must be, or there could be fibers going in this direction, but then maybe there's more like water here, or there's not so much information here or up here. And what the tractography now does is to start at one point, and then we just go from voxel to voxel, right? We always look at which direction does it go, and we basically move through the tissue and take and record this, and we get a streamline track. And so that, and here we stop maybe because there's more water and it's not clear where to go, so we stop. And so that's a basic concept. We could do this a lot of times and maybe add some probability function, and then it's called probabilistic tractography. We could also, it's not that. Then maybe sometimes, because of probability, we end up some very different place, right? So because sometimes we take a different route. And then we could draw a heat map of what is connected or not.
May I ask a question here?
Sure.
For the tractography, one point I never understood before, probability like this, how do you decide whether water is flowing in this direction or this direction?
Yeah, so basically what you do is to add noise to the data. That's simple. So you add, you don't take always the main direction, but you add noise. I have on the slide of that. Yeah, this one. So this is probabilistic tractography. So you may have a really, so let's say, take this one. You have good information that the tracks go like this, but you also add some Gaussian function to it, so you are allowed sometimes to go like this, or like this. So probabilistic tractography is really adding noise to the data, I think. So that's, nobody describes it that way, and they all have fancy terms, but it's basically not always taking the best, but sometimes taking a detour.
So this really comes down to probability.
Yeah.
No.
Yeah. And I mean, it's called probabilistic because they sometimes start here and then do 100,000 streamlines, and then you have a probability of being connected to this point that you can, the sum would add up to 1, and you would you could say something about how likely is it to end up here. But, yeah, and so okay, so all right. So this is histology. This is in vivo. So with good data, you can also reconstruct good fiber tracts. But Yeah, about this deterministic versus probabilistic tractography, it's always the first question that people ask you, and if they do, you can already maybe say they are no experts, because it's the typical thing that clinicians ask, or people that have heard about DTI but don't know much, and they often ask, is it deterministic or is it probabilistic? And it's often, I think, the wrong question to ask altogether. Much more important than that is really what model you put here. Right? What model do you model your voxels with? Is it a tensor? And that's really old school. Nobody does it anymore. I still explain it this way because it's the easiest. But usually we have much more complex models here that have multi-tensors or Q-ball or Q-shell imaging, whatever. There are so many things, spherical, deconvolution, and so on. So that choice, in my view, is much more important than whether you do a probabilistic or deterministic tractography. So, which is actually essentially whether you add noise or no noise to the data, right? So, I think the differentiation between deterministic and probabilistic is important, but it's maybe not the only one. It doesn't describe really what you're doing. Other options that you can characterize it is whether you do a local or global tractography. Global tractography are rarer and more rare algorithms, and I'll show you one before that try to model the whole brain directly. So they don't start as we've just done, started some corner and start there, but they start everywhere at once. And then you could also think about whether the data is stored as streamlines, as we've just seen. We could record these streamlines and write them down, and write down lists of coordinates of the streamlines, or rather maybe these probability maps. But even much more important is really how do you model each voxel? Right? Do you fit a tensor, or do you fit a multi-tensor, or do you fit a ball and stick, or a cue ball, or whatever? So that, in my view, that's the most important thing. Not so much deterministic versus probabilistic. You can then use it. This is actually from my medical thesis, so very old stuff. But you can then use, have regions in the brain and connectivity tracks between them, and look at anatomical connections. General problems or shortcomings in tractography are huge, so it's really not a good method. Think about it like that, but it's the only method we have if we don't cut up the brain. But it's not a good method, really. It's terrible, actually. But still, so one problem is crossing fibers. So if you have regions where fibers cross, then in the end it's hard to resolve it, because you will have diffusion in two directions, and you don't know, you cannot really see that well, right? It looks more, if you have diffusion in all directions, it looks like water again in the end. So that's one big problem. Then what people call the length bias is that if you start here and track through the brain, then it's just more likely to get closer regions, because at some point the algorithm stops. And just because the probability of stopping adds up because of noise and everything over the length, you get more shorter connections more easily. In my view, also, this is a big problem that we can maybe even solve partly with global tractography. Then one is the seed bias, where you think about, you place one seed here, maybe across subjects, and you want to know what's connected to it. But in some subjects you are more into white matter, and some you are more into gray matter. And then you get more fibers in the ones where your seed is more in the white matter, but it doesn't have anything to do with connectivity in the brain. So that's also one typical bias, and so on. Cross-sectional area invariance means that you have sometimes bundles that have really a lot of fibers, but they're only in one voxel, and you cannot really resolve this in DTI. If you have bigger bundles, then you get usually more fibers. Error summation is basically that at each point on the track you can make a mistake, and if you are up here, the error sums up higher because there are more possibilities to make errors. And I think a solution to some of the problems, but not all of the problems, is really global tractography, which tries to, in one step, in one computational step, reconstruct the whole connectome as one. And so it also tries to model the whole MR signal in one step. So you don't have any tensors, you don't have anything like that, but you just have your diffusion signal and you model the tracts, and you want the tracts to explain the diffusion signal. So that's research from Freiburg. It's called Gibbs tracking, and how I started with diffusion MRI. The concept is that you have these particles, so cylinders that move around in the brain in a simulation, and then you want them to align to tracts in the end, right? So they polymerize to fiber tracts. So in the first step they move around, and you have this temperature that you can model, and then you decrease the temperature, and at some point they align to tracts, and they want to explain the diffusion signal. So each of them emits a local diffusion signal, and in the end you get a global diffusion signal, and compare that to the empirical diffusion signal. And you will have these sticks here. You have the distance between the ends of these sticks, and this distance needs to be minimized. So that's the what they call the local energy. So they want to minimize the distances between the sticks so that they polymerize. And then they have the global energy, where you compare the data, empirical data with the model data. This was published in 2011. I think it's a really great concept. So you can see also that it finds a lot of fiber tracts, so it was validated that way. It's really good for crossing fibers, which resolves that. That was really well. And also, if you compare the FA mass, I haven't introduced this, but this is basically how much spheres-like is your data, so how much water-like was. You have more water in the crossing region, so more, non-directed diffusion. And in their reconstructions of the fiber densities, you don't have that problem, so you can really see that tracts are resolved well in there. But that's just one option. So there are tons of software tools. I think most popular is FSL, which they really say probabilistic tractography is great, and they have a small and 6 model, and I think there's a lot of documentation out there to use FSL. So it's a very common tool. Then there's the DTI toolkit and TrackWiz from Harvard, from the Martino Center. I think they had this one controversial but very influential science publication with orthogonal tracts and so on, and the TrackWiz software is really nice for visualization. So you can use Lead-DBS to export to TrackWiz format as well. Then I think under maybe the more expert people, Matrix is really popular. So a lot of people that are only doing diffusion MRI tell me it's the best software. I don't know if it's true, but it's actually pretty good. Also nice visualizations and so on. Then this is a really outcast. This is the Freiburg data that I just showed you. So nobody uses this except me. No, I mean a lot of people use it. And inside the methods people, it's very known. So I think at the MICA and the methods meetings, everybody knows the tranquilizers out of it. So they actually included the Gibbs tracking algorithm also into a matrix now, so it's slowly becoming more popular. Then for people that like MATLAB, Explore GTI is a really good software. It's developed by Alexander Lehmans and Derek Jones, but I think more Alexander Lehmans, and he's really a great expert, and he really takes things seriously. So with that toolbox, there's also a lot of quality control options, and you can really explore, as it says, your data. And it's all in MATLAB, so it's also easy to use, well documented, it's free, so on. Then there's DTI Studio, and I have not used that, but Susumu Mori was one of the first people who really did tractography and the fact tracking and so on. And then there's a German product which is also pretty nice and easy to use. It's called K Diffusion. Also has a Gibbs tracker adaptation, so also has this global tractography. And then what I really like, I even think it's the best neuroimaging software ever written. It's called DSI Studio. It's so easy to use, so fast, and beautiful visualizations. So it's good for beginners, but still it's also a great algorithm, I think. By Fang Cheng, also great support. He answers to emails very well. So I can really recommend this as well. But I don't want to make any recommendations myself. What people have done though is to compare tractography algorithms. So there was a really influential big study that came out as a preprint early by Klaus Meyer-Hein. And if you know the diffusion MRI world, these are all the big names. So nearly everyone is on here. And they actually had a lot of, so everybody tried to reconstruct tracts. And what they found is very frustrating because they found that there are 4 times more false positive tracts than true positive tracts in the average tractography. That's why I tell you it's a terrible method. Don't use it. No, but really, we have to be careful. We cannot use DTI or tractography to identify new tracts or so. It's just not the method for that. We need animal work or histology for that. But we may maybe use it for some interesting things in the human brain as well. And it was then published in Nature Communications, so now a really well accepted paper. And in that challenge, the BSI studio method just achieved the highest valid connection. And it was 96 methods from 20 research groups. So and with that, I think it's the perfect tool for clinical DTI, so for clinical applications, because valid connections means not so many false positives as the others. There are, of course, again, a lot of false positives, but it's in this competition, it was the winner somehow. And especially for clinical use, I think we don't want a lot of tracts, but we rather want true tracts, or as many true tracts as possible. So that's why we, and before that there was another challenge, the 2009 NeuroCup, where the global tracking won. It was the winner in the Filler 2011 paper. That's why we first started to implement global tracking into Lead-DBS. And then Marco Reisert created a new global tracker, which is called the Meso tracker, which we then also included. But then we also read the Meyer-Hein study that I just showed you and thought, okay, we need to have DSI Studio in there as well. So basically, these two are the methods from big open competitions. So they are not the worst choices that you can take. This is a paper, a figure from the new Lead-DBS 2.0 paper, where we just had one patient use a very old school DTI method, and then we used global tracking, meso tracking, GQI, but also used normative connectomes to show the whole connectome, but also then the selection of the different simulation parameters. You see there's a big difference among methods, right? So it's a terrible method. But maybe one thing to also note is that if you use Lead-DBS or lead connectome to do this, then it will always create a whole brain connectivity and then select the fiber tracks. So it's different than other software. It will always, no matter which method you choose, it will always first reconstruct all fibers, naive to where you look at, and in the second step it will select the fiber tracks. And I think that's the way to go. It has a lot of the same problems that we discussed before, like seed bias and length bias, but some of them are a little bit attenuated or a little bit accounted for, because first you track all fibers without any, you don't know the question, you just want a reconstruction of the whole brain, and then you select the fibers. So you don't have, so if you instead would track from the VTA, the algorithm would specialize on the VTA maybe. But if you do it without that first and then select them, there might be a bit advantage. Okay, so the idea would now be to have a hands-on session, and I would just show you how to reconstruct the whole brain connectome in Lead connectome, but we don't have, so our data set we gave you doesn't have diffusion MRI scan. So you can either just follow me while I do it and look at it, or if you have diffusion MRI data, then of course you can also do that. Okay. DTI.eval and DTI.evac. And it's called DTI, but we don't really do diffusion tensor imaging, so it's a historical, it should be diffusion MRI actually. But what you can see, it's also not, in this case, not a good scan, not at all. It's a clinical scan, it's even with electrodes inside. But for this demonstration, it doesn't matter now. But usually, of course, you would want to have a better resolution. So here we have 2.5 millimeter resolution, and we only have 38 diffusion directions. So it's really not a really good data set. But just to demonstrate it for you, I think that should work. And inside of these DTI eval and evac files, they are automatically generated if you import the DICOMs with GTI, with Lead-DBS, but they account for the diffusion vectors, so gradient vectors and the values. Okay. So all you basically need to do is put in these files, and then the rest should work. And what we can do now is to open this patient lead connectome. So it will be loaded in a in the second app that we have. The other option is to just type in lead connectome and press enter, and it will just open the same folder, same app. The third option would be to just use Lead-DBS and just click on, check this one, this option, and open the settings dialog, which will then have all you need from lead connectome as well. So will it be the same in the end, but if you have a rather DBS patient with electrodes, it might make sense to just stay in Lead-DBS and run the connectome as a plugin, and if not, if it's just about connectome data, maybe you prefer to just open lead connectome. But it's going to be the same in the end. But we'll do it right now, so like this, we stay in Lead-DBS, but we have lead connectome open, so I can open settings, then I get this window, and I can just say basically perform fiber tracking, and I will use the generalized Q sampling method. And I also want to normalize the fiber tracks into template space. And maybe that's it for now. And I say save and close, and I say run. And it now will basically pre-process everything. And in this case, it's already pre-processed, but in fact, it's already done tracking. It's incredibly fast. Diffusion DSI Studio is, as I said, the best neuroimaging software ever written in my view. So we already have sampled the whole brain connectome right now. And as I told you, it was still the winning method. It's not a bad method, so don't be fooled by its speed.
So what kind of pre-processing has been done?
The pre-processing, that's a good point. We don't do a lot of pre-processing, but we do GIPS unringing and registration to T1. That's all.
Okay.
And
so
we don't do
motion corrector, eddy corrector.
We don't do that. Yes.
So
We
we did process it
yeah.
before.
You could do that before, or you could not do it. Yes. And I think there are even some experts that say it's not always good to do everything, but I agree. So we want to add these tools as well. Yeah, I think Ningfe is thinking about putting eddy and so FSL's eddy and top up into it at some point, but we need to find the time. But if you want to do that, you should run it in a different, like in FSL, and then put in pre-processed data. Okay, so now we've done that, and now it's normalizing the fibers, which takes longer than the actual tracking. And what we can also already see is, so I sort this by modifications. We have the fiber tracks that are stored here, and these will now be converted to track-width format as well and normalized into anatomical space, into MNI space. So in the end, what we will get is ftr.track file, which is this one, and we can open that in track-width and just for visualization. So we can do that now. So TrackWiz is again this software from Boston, from the Martino Center, and we will have that, but it looks, let me see, can show everything.
And you shut down this project, so field.
Yeah. Here we go. Okay, so we have these 5 tracks. We sometimes, as you can see, we sometimes have problems converting to TrackWiz format in a proper way. It works pretty well for the MNI space, but it does not somehow in native space. But you can still look at, do the tracks make sense at least? This is so weird. And you see, for example, there are some issues. So here, for example, there's a gap. So that might be because it's poor resolution, or because of the tracking mask is not optimal. Except that it looks okay for such a clinical data set for me, to me. And let's see if here, now you see the normalization. So it goes from B0 space over to anatomical T1 space and then to MNI space. So this is just gives you a rough estimate that this worked. It didn't somehow fail. And now we also have a normative connectome of that patient, which is in MRI space, and that's inside connectomes, diffusion MRI, and then WFTR. And we can visualize that a bit more easily in TrackWiz. So it's the same data, but just mapped to MRI space. Say we want to show everything. Now the orientation is correct and everything. We still have this gap, of course, but if we say open load image and take maybe the T1 of the MRI space as a backdrop, we can also see that this is now registered to MRI space. Right? You can see that it's in one of the spaces. Okay. Then maybe as a next step what we want to do is we also want to see a connectivity matrix of these tracts, right? So we want to maybe see, take the AAL, for example, which is a typical parcellation method, and we just want to visualize the connectivity matrix, and we also want to compute degree centrality, which is a simple graph metric. So we can run that now, and it will now take the parcellation scheme into native space. So we see Connectomics now. This is the new folder that was created today. So we have, it's called after the parcellation scheme. So we have AAL, and it will now warp the AAL parcellation into native space and use it to, based on the fiber tracks, to create a connectivity matrix and a degree centrality. So yes, iterating through all of these fibers, 200,000, and yeah, creating this structural connectivity matrix. Okay, here you have the matrix now. That is based on the entries of the AAL parcellation. And now this is the degree centrality map. So for each parcel, so it's actually strength centrality. But for each parcel in the AEL parcellation, you have the number of connections it has to other parcels. And maybe I mentioned before that we have quite many of these parcellations in Lead-Connectome, and if you use them, I think it actually makes sense to read the papers about what they are, you know, or try how were they defined, which one is the best for me. There's no good answer. A lot of people of course. Yeah. Yeah, that's a tough question. I think with tumors, you cannot really use Lead-DBS to do that. Because, so you would need to use, for example, a normalization scheme manually with ants or so. There's a good documentation to normalize grains with lesions to draw a mask and say, don't use that and so on. But you would need to use custom tools. It would not work with Yeah, you can. Yeah. So maybe these parcellations, as I mentioned in the beginning, look at also under help support, knowledge base, atlas resources, and then cortical atlas parcellations. That's this page. So you will have information about each of these, you know, parcellations, how they were made, and where to get more information, or which is the paper to cite if you use them, or to read about them. Right, so there are quite some different options that you can use. If you want to draw yourself, these are stored inside your Lead-DBS folder, again in the M&I folders, which we have seen a few times now, so in template space, M&I. Then under labeling, right, there they are. And we have used the automated anatomical labeling one, and it's always a text file and a Nifty file. The Nifty file will just be the voxels drawn, and the text file will be that all region with one will be this region. So it's very, very simple format. Right? You could easily make one like this.
And how do you get from there to the connectivity profile that you are showing us?
You mean the connectivity matrix, or?
No, the profile for the connectivity with the colors. That's somewhere else. So that's actually in the mapper, which we will cover in later. And plus scripts, so you would need actually custom scripts to do it. But it's all in here, it's easy. So it's basically AAR map, and then you feed in your data. Okay, but so this is a bit different than what I've shown you before. This is really processing patient-specific data. Right? So it's not using normative connectomes. But of course, you could also use these connectivity estimates in your patients.